Okay, just give us 2, 3 more minutes. We'll proceed. Ah, yeah, yeah. Yeah, I have. Okay, there might be a little bit of things going on behind me, but I would like to start the colloquium. So it's a pleasure to welcome all of you back to the Wednesday Colloquium Forum. Um, Please excuse a little bit of a delayed start, but nevertheless, I give you the 2023 Molecular Ecology Prize winner, Professor Ramakrishnan, who has come all the way from NCBS. So this is a fantastic occasion. The last time she visited was almost 10 years back. She gave a colloquium here in 2012. And Uma is pretty unique in her work in ecology and has been, following on tigers for a long time in India. And her work has had an international recognition. Uma actually background. It's important that we recognize the where she's coming from what was the basic training. Okay. Uma did her bachelor's in science in physics, chemistry, and mathematics. And then she went on to do her master's in biotechnology. After which, and this is all from Bangalore University. Then she moved to UCSD for her PhD in ecology and continued her work in ecology in Stanford as a postdoc before coming back to India and joining NCBS almost 20 years back. And now she's a full professor there. And her work, of course, as she will talk about few aspects of it, has garnered lots of prizes, and one of them was the last year's prize of the Molecular Ecology Prize, and the other one is the Parker Gentry Award. Both are international recognition. Clearly establishes her worldwide, her work and her interest and excitement to follow large mammals and the genetics of it. So I welcome Uma. Thanks. Is it on? Yes. So- Sorry for the delayed start. Sorry. No, it's okay. I'm not on Edu Room. Yeah. Yeah, that's fine. I can change the slides. So, oh,
It's a very nice statement from this. I can easily choose.
Okay, so my name is Uma, and basically, I've been with my team of amazing early career researchers. We've been trying to understand more about tigers, but from a genetic perspective. And actually, I don't know whether you know, but today is I think it's International Environment Day or something like that. So it all worked out in some sense. So what's really amazing is we forget, but we are surrounded by like really beautiful biodiversity. It's also kind of crazy because if you look at it in terms of biomass, 96% of biomass on Earth is humans and cattle, basically, and only 4% is wild. Yet there's such like amazing biodiversity, whether it's like colorful butterflies or amazing habitats like in the Western Ghats, or this really charismatic leopard, which basically surrounds us. And this biodiversity is not equally distributed across the world. I love maps, and this is a beautiful map showing distribution of biodiversity across the world. And you can see, of course, that there's a concentration in the tropics. But for example, you also see high biodiversity in mountain ranges and so on. So for a long time, people who studied biodiversity, including people like Darwin and Wallace, kind of wondered about how biodiversity was distributed. Even before, today we think about biodiversity more in terms of a crisis mode, but it has fascinated scientists for really, really long. But does it matter? Like, of course, it's very beautiful and we want to understand the patterns, but how much does biodiversity really matter? So I think that in the last 10 years, yeah.
When you draw a map like that, is it number of species?
Yeah, that's species richness. Yeah. To the best of our understanding, we have very poor understanding of most distributions, but yeah. And I think this is something which has come up a lot recently, even to the extent of, it's almost become revolutionary, this desire to protect biodiversity. For example, someone like Greta Thunberg, going and actually, going on strike every Friday. That's a big thing, right? For something like biodiversity or climate, I think people are beginning to wonder that it matters. And I think in India, for example, I personally feel that a lot of the challenges that face the country are to do with environmental degradation. And we are seeing these in terms of floods and heat waves. And this is something, the connection between climate change and biodiversity is very strong. I'm not going to talk about all of that today, but just giving a preface that it does really matter. And we're basically losing biodiversity very rapidly. That's the IPBES report. And for example, some studies suggest that 1 in 4 mammals, for example, is threatened with extinction. So that's all nice. This all sounds like, what you read in the newspaper. But how does that connect to us as scientists, right? So of course, as a scientist, and before we go there, this is just a picture of India, 1700 and 2000. You can see that forest cover has gone from 60% to 9% in 300 years, and villages and agriculture have gone from 20 to 78%. And you might have seen this recently, the chimpanzee. Sorry for that. This is my slide. But basically, as biologists, how do we understand extinction? So what does extinction depend on? So a lot of studies in the past have shown that extinction depends on population size. So when you have large populations, probabilities of extinction are really low. With small populations, probabilities of extinction are high because of stochastic effects, environmental stochasticity, demographic stochasticity, and so on. Similarly, this is data from butterflies, but similarly, when you have lots of small patches which are isolated from each other, like here, you have many isolated patches, the chances of extinction are high, but when there are many connected patches, the chances of extinction are lower because there's potential for recolonization, and what happens in one population can kind of be offset by others. Empirical data, yeah, from butterflies mostly.
10⁶. What data do you use for that? Time to extinction of the scale of 10⁻10.
So this is basically based on long-term studies, and it's in terms of generations also. So basically, since they have shorter generation times, you can actually 10 to the 10th, yeah, but I think it's a, so this is basically probably a projection. I think this is definitely empirical data. This is probably a projection based on simulations. When you see these isolated populations, and if you don't allow recolonization, then you have potentially extinction. But it's just a probability, right? In the sense that out of 1 million populations, something goes extinct, something like that. Okay, so the other thing that impacts extinction is genetic diversity. So, for example, this is also old data showing that in butterfly populations, despite correcting for environmental and demographic stochasticity, populations with low genetic variation, heterozygosity is, there's a probability of 2 different variants. You find that extinction probability is high when heterozygosity is low, and over time, when you have if you have higher heterozygosity, probability of extinction goes down. So this still seems very heuristic, like very correlative, right? Why does this happen? So one reason is small populations are also subject to genetic stochasticity. So by chance, certain genetic variants which are not adaptive, which are maladaptive, might increase in frequency, and this pushes a population towards extinction. The other thing that happens is when you have small populations, you have inbreeding. Individuals who are related carry a similar load or repertoire of bad mutations. And so over time, these mutations come together and you have what's called inbreeding depression. And then again, that decreases fitness and potentially causes extinction. So this is all very nice, but like, how does it connect to tigers, right? So that's the question we are trying to answer. So tigers are an interesting model for extinction because first of all, we've already seen extinction. This is the historical range of tigers. You can see that they were actually present in a large area. And the darker red is where they're present today. They've already gone extinct, for example, in South China. They've gone extinct in the Caspian region, also potentially extinct in island Southeast Asia. But basically, they still exist in a place like India. But we've seen that India is very fragmented, right? And we've lost a lot of habitat. So, now what we are left with are lots of small patches of populations. So what are the populations? What are the trajectories of these populations going to be? Can we understand something about
Can
this?
I just ask a quick question?
Sure.
Are you only showing, is this the only places in the world that tigers are found?
Yes. No. So basically, tigers are Asian. So for example, lions are African, sort of African origin. Yeah. So tigers are definitely Asian in that sense. Okay. So what I'll do today is a lot of stuff. Maybe I won't get through all of it. Is the first questions we asked were kind of descriptive. What are the populations? What are the genetic units which hang together, right? And if you have populations which are larger than just one protected area or one place where they live, what sustains these connected populations? What's going on today which could probably impede connectivity? Of course, there are already isolated populations. What happens here? We talked about 2 processes, drift and inbreeding, and basically a little bit about very recent work which we've just finished and what we're going to do in the future. How do we do this? So you have to get biological material from tigers. So we don't catch tigers. It's too risky and very difficult to get permits to do that. So we mostly walk through forests. And the good thing is they're cats, so they mark their territory. So they defecate and they spray urine, basically to give signals to the other individuals that this is my territory. And we just follow along these paths and we either pick up like swab these fecal samples, or in other cases, we find shed hair. So they also scrape and mark. And if you really lay down on the forest floor and look, you'll find tiger hair in those scrape marks. So basically, we just bring this stuff back to the lab, extract DNA, and then recently we're doing a lot of genome sequencing, and I'll talk a little bit more about that. So just to also say that there are two key innovations which really allow our work to happen, and this is something which we don't acknowledge often in science, but basically this process called PCR, which allows you to copy DNA, is critical because the amount of DNA we get is very less, and we need to be able to make a droplet of DNA into a swimming pool of DNA, and PCR allows us to do that. And more recently, the next generation sequencing, which is massively parallel sequencing, basically broke Moore's law in terms of the cost, and it became much easier to also sequence bad DNA, small bits of DNA. Like all the DNA we get is like a torn up book. Your genome is a book. We're accessing stuff which is bad and in low amounts. So what we still, of course, need is really better abilities to compute and to do analyses. Analyses methods are also growing substantially. So this is just very quickly what's happening globally. So this is tigers across the world, sampled from all these various locations. And this is a PCA plot, basically just shows you which collapse by like there are millions of SNPs or single nucleotide polymorphisms or differences between all of these individuals. And you can basically see, for example, that, the so-called subspecies, Amur tigers, Sumatran tigers, and so on, kind of cluster with each other. And basically, say, Indian tigers, that's called Bengal, they cluster together. What's really interesting is, if you look at these tigers from the northeast, basically Assam, they show some admixture. They show these, basically each line is an individual, and the color is, what is it genetically? What cluster does it belong to? And you can see that these northeast individuals have some elements of the other cluster. So probably this is a mixing place for tigers. What's even more interesting is if you look at genetic variation, the amount of variation, this is just like a rarefaction curve. Indian tigers have way more genetic variation together, all of them, right, compared to any other of the subspecies. And also, though, this analysis, you're going to see many of these plots. These are what are called runs of homozygosity, or stretches in the genome which have no variation at all. So though all Indian tigers have a lot of variation, they're also very homozygous individuals, which means that maybe this fragmentation of habitat is already having effects on their genomes in terms of inbreeding, but we'll talk more about that later. So it's it almost seems contradictory. Together, Indian tigers have a lot of variation, but individuals are potentially inbred. So if they have so much of variation, what allows them to be connected? So this is some work we did some years ago, where we looked at, this is basically MP, Maharashtra, Madhya Pradesh, and so on. And Prachi walked thousands of kilometers, collected tiger scats, and looked at how genetically similar or different they are. And then we basically use this conductance or resistance-based models, where you have each protected area is outputting current, which is tigers, and you have resistance driven by the landscape. So for example, if you have a lot of forest, that they'll coast through that maybe. You have big cities, they may not. But what we do is it's a correlational approach. You optimize the resistance that matches the genetic patterns. And what we find is basically that overall populations are connected, but high traffic roads and high density human settlements, basically cities, are barriers. Otherwise, it's not a problem. Agriculture, not a barrier, no problem, which is good news, except that while we are doing this work, this is Kanha and this is Pench, just at the border of Maharashtra and Madhya Pradesh. This highway, which was 2-lane, was going to be expanded to a 6-lane highway. And luckily for us, some citizens challenged this decision in court, and our papers were used in court basically to petition for this overpass. And the argument was that by building this, expanding this highway, we're going to create a barrier. And we actually did simulations which showed that if this becomes a barrier, extinction probabilities of that whole landscape increase. So basically, this work allowed for this overpass, which is pretty interesting to think of, a paper allowing for something in cement. And what's really nice is this underpass is working. So these are camera trap images. That was 2019. And these are camera trap images, very ugly underpass. I wish they would make it look nicer. But here's a mother and her cubs, males, even ungulates, gore. This is Asiatic wild dog, dhole, all moving under the underpass. So basically that whole area between Kanha and Pench stays connected today. So even as we talk about connectivity, this connectivity itself is threatened, right? And one way it's threatened is by roads and expansion of roads, but there are solutions like these underpasses. Yeah.
This wonderful stuff. Just go back to the resistance current plots that you had. Could you just explain a little bit? Did you also, is there a height in that topography, like in that map?
No, there's a little bit, but topography does not correlate with resistance, does not predict resistance.
Okay.
It seems like basically resistance is very, like high traffic roads and high density human settlements have very high resistance, like 1,000 times, but like resistance of agriculture is 10 or something like that, relatively scale.
But is a data set that you have around that area? This is all connections that you have are data sets from constructed from data sets?
These are the samples, right?
Oh,
yeah.
So you
and then basically you look at genetic distance as realized flow, and then you correlate it to get some prediction of,
You have cells which are connected by resistors.
Exactly. Yeah.
Just electrical resistance.
Yeah, electrical resistance. Yeah. What that resistance is depends on what, like, is it farm or is it what is it? Okay. So now, basically, that's not the only thing which impedes connectivity. I'm sure all of you read the news and you must be seeing what's happening in Chandrapur. There have been so many attacks on humans, right, by tigers. So tigers have recovered in, at least in central India, definitely in Maharashtra and Madhya Pradesh, and they are territorial animals. So the parks are so small, there's not enough space for them. So they might end up moving out. And that's what we want, right? We want connectivity. We want them to move across these protected areas. That's what I said earlier. But when they move through these landscapes which have people, they will also potentially end up with conflict, right? Either killing livestock or killing people, right? And that's not acceptable. To kill people is not acceptable. There's immediately backlash against the carnivores. Actually, India, people are very tolerant, but it's not a good thing. This is a montage of camera trap images from outside Bandhavgarh. And you can see this female here. Here she is at around 9:15 p.m. Here she is with her 3 cubs at 11:30 p.m. Here she is. These are, by the way, like 2-month-old cubs, 7:40 a.m., same spot, 12:46 p.m. Okay, and these women are walking by. I mean, these pictures are probably not surprising to you living in Mumbai, where you've seen all those amazing pictures of leopards from Sanjay Gandhi National Park with the Mumbai skyline on their, like, at the back. But this is not a good situation. So what we wanted to ask is, okay, conflict is happening, but can we actually use these genetic tools to understand something about it, about the patterns, who is involved? So the general narrative is, there is a problem animal, take it out, okay? And that's the narrative that most of the bureaucrats use. So we thought we look at this problem a little bit more, and this is again a camera trap video. Yeah. So here, these are 2 cubs, and you can see there's a cattle kill there. And I don't know whether you can see, but they're like batting it around a lot. And I don't know whether any of you have cats, but cats play a lot with their prey. And so this is basically what we used, because when they do that, they basically leave traces of themselves on the prey, right? So for example, this is a bite mark. So usually they kill at the jugular, they bite, and then there is saliva where they've bitten. Okay, there are also a lot of lick marks. Cats clean their prey, so they lick it, and there again is saliva. So what we did is, this is Himanshu, he's quite a crazy character, but we sampled more than 100 cattle kills in this way by just taking a swab and rubbing it over that lick mark, just shoving it into the bite mark, just to see what will we get. Sounds crazy, right? I thought it sounds crazy also because this kind of sampling is very crazy. The kill is there, you're kneeling down, and the tiger could be anywhere around. So it's quite risky. So what did we find? I'll just summarize these results for you. This is Bandhavgarh. Oh, sorry, I went ahead. Ooh, went very far ahead. This is Bandhavgarh, and this is the number of kills across space. So you can, of course, see that there are spatial pinch points, some places where there are more kills. Now, we've not done a detailed enough study to say why. Maybe there are more cattle here. We don't know exactly why. But this is the really interesting result. This is the number of... These are only cattle. Only cattle. Yeah, so people put their cattle, they're not supposed to, but there are cattle in the core as well. Yeah, so basically this is the interesting result. This is the number of individuals at 1 kill, 2 kills, 2 different kills, 3 kills, 4 kills, 5 kills, 6 kills, and 7 kills. So if there was a problem individual, all the kills were because of one animal, we should have seen a huge frequency here, right? Or a few individuals doing lots of kills. That is not what we see. The population of Bandhavgarh is about 80 to 100. So more than half the tigers of Bandhavgarh are coming and doing cattle kills. So now, I mean, this is not probably a good result. Just as individuals. So basically from each kill site, we identify an individual, right? And when another kill site, is it the same individual or a different individual? So basically this is 40 distinct individuals at 1 kill each.
Yeah, within what time?
Oh, this was all done over 3 seasons. So like about 8 months, something like that. But so we did a lot of other stuff to look at how effective this method is and so on. Can it actually be used to identify individuals? And basically what we found is that at 86% of the kill sites, we can identify genetically some tiger, right? So this is nice, but actually we don't know. Yeah, sorry.
Sorry, one question. So why is there no data in the southern region?
Sorry?
Why is there no data in the southern region of your map?
Oh, here?
Yes.
Yeah,
the
so basically,
south.
yeah. So basically the thing is, we give out these cards to all the villages around. And what they say is basically, if you have a cattle kill, call us, and then we'll come and sample. So of course, there are going to be biases. Sometimes people don't call us, but we also got records from the department about where there were kills. And overall, it matches. Like, it's not like there's a massive spatial bias in terms of our sampling.
To understand the map.
Yeah.
Okay.
The different size things are number of kills or number of individual tigers?
Number of individuals at that site.
At that site.
Yeah.
So then I should expect it to correlate with the density of tigers and not with the density of prey.
Definitely. Yes. So that is something which I'm kind of skipping over the details. A lot of people have suggested that cattle kills happen because there is no wild prey. But what we have found for tigers, I mean, we have not proved it. What it seems to suggest, and some other data which we collaborated on for snow leopards, suggests that when there are large numbers of animals, irrespective of whether there's wild prey, there will be cattle killing. So basically, probably these populations are beyond carrying capacity. Also, cattle are easy kill compared to a gaur or, a big animal. I already said this.
You have similar type of pain, because Bandhavgarh is clearly very, right?
No, we do not have similar data for painch. But what we are thinking about is whether we should do more of this. For example, we actually got a call from Chandrapur Tadoba, where they want us to. So the problem, of course, is that I know that this is a different tiger and this is a different tiger at these two kill sites by their DNA, but I don't know who they are, right? So for any kind of a management or an intervention, I need to say this is this tiger, right? So that they can catch it or not or whatever. So just to show you an example, so this would require a database, like an Aadhaar card for tigers, where you have the genetic kind of fingerprint or whatever you want to call it of all the individuals. And that's what Tadoba, for example, is asking us to do. But actually, this project took us like 2 years. That is too slow for management, right? They want to know till today, in a week, they want to know whom should they catch. And we don't work on those timelines at all. So just to show you an example of this funny looking bird, it's called a kakapau. It's found on New Zealand, in the islands of New Zealand. And here, every single bird is genotyped and they do it in the field. So there's these small sequencers called nanopore sequencers. They take them into the field and they do like identify. So they have basically libraries of individuals, and it's basically, is it this or that or this or that is what the output tells them. So I don't know whether we'll ever go that far, but just to show you, these are the kind of applications that people are talking about internationally. Okay, so now I'm going to talk a bit about some already published work, but really fun work about isolated populations. So these beautiful tigers, I don't know whether any of you are on social media, they keep showing up. They keep showing up more and more than they were even before. This is a black tiger or a pseudo-melanistic tiger. Why not melanistic? You must have heard of black leopards. We call them Bagheera melanistic leopards. They are black, completely black. So they have no other color. But these guys, they have the three colors of tigers, orange, white, and black. But the stripes are irregular. Okay. So actually, this is an interesting kind of phenotype, because it turns out that many people have been studying these patterns in cats, and they found that a specific gene called TACPEP is involved in development and mutation. When TACPEP is normal, the stripes are laid regularly. When TACPEP is abnormal, there's a mutation, the stripes are all over the place. Okay, but this is for cats. So anyway, we got lucky in some sense. People were shouting about this phenotype. They were like, what's going on with this tiger? Why does it look this way? So we said, okay, maybe we had a hint. It might be a TACPEP mutation. And so luckily though for us, there was a litter born in Nandankanan Zoo. This is Nandankanan Zoo. It's in Bhubaneswar, where there was a white mother, a white but pseudo-melanistic daughter. So she is white, but her stripes are all over the place. A pseudo-melanistic son and two normal sons. So we built a pedigree, right? This is a 3-generation pedigree where we sequence genomes of all the individuals in the pedigree. We looked at this specific gene, TACPEP, and we found a mutation from histidine to tyrosine in a very conserved region. Basically, parts of this gene change easily and other parts don't, they're conserved. And in that conserved bit, there was a mutation, and it's basically an association study. So individuals with these mutations were 200 more times likely to be pseudo-melanistic. That is in the zoo. So now we have to go back to the field. So basically, Vinay went back to Simlipal, where he walked up to 1,000 kilometers along these trails, just picked up scats. He was there for 3 months. He never saw a tiger. Very low density park, right? And basically brought them back to the lab, extracted DNA, identified tigers, maybe we picked up a leopard or something, identified individuals, and then typed this mutation. So basically, this is the black. It's a recessive trait. So an individual is black if it has 2 copies of the mutation. And what we found was that the frequency of, so basically we found only 12 tigers, and 4 of them had 2 copies of this mutation, which translates to a frequency of almost 0.6. Okay, because it's 0.6 squared, you need to have 2 copies of the mutation to show it. So maybe we missed it elsewhere, maybe it's there and we just missed it because it was in heterozygous state and the tigers look normal. So we actually did a lot of horse trading with all kinds of people and managed to get 400 individuals, tiger individuals from across range, mostly from India. And basically the mutation is absent or rare. I mean, of course, we can never say it's absent everywhere else except Simlipal. So what is it about Simlipal? Of course, everybody immediately said, adaptation, the forests in Simlipal are dark and this dark tiger must be doing really well there. So we can't say that for sure that it's not selection, but our results show that it is consistent with drift or just isolation and changes in frequency by chance. So basically, first of all, we needed to show that Simlipal is small and isolated. That is the place where drift happens, right? Small isolated populations. And basically, this is just a PCA plot which shows you that compared to neighboring areas, like this is quite a large area, right? Tadoba to Satpura, Kanha, Bandhavgarh, and so on. Simlipal is genetically sort of on its own and distinct. We also said there were only 12 tigers, so it's obviously small. And also, individuals in Simlipal are quite related to each other. So this is relatedness, pairwise relatedness between individuals. Of course, individuals in central India and southern India are not so related. But Ranthambore, I'll talk to you about it again in a bit, is a highly related. Individuals are inbred here. And Simlipal is kind of on that same order on average, about almost SIB level related individuals. So, yeah.
Since in individual pockets there is not much variation.
Yeah.
Is it even possible to move them from?
Yeah, I'll come to that in the end. Yeah, so that's what we are. In fact, just 2 days ago, I got a letter from Odisha state saying, we really now, given all of this work and we want to try and think of genetic rescue. How do we bring in individuals to this population? And so, of course, they asked me, so where do we bring them from? That's a whole another 5 years of work. But anyway, that's hopefully where we'll get to eventually. So this, yeah.
So even though you have, for instance, the place where you said there's much greater density.
Yeah.
Right,
Central India and all that. Yeah.
No, but you use a specific term that's your
Carrying capacity. Yeah.
You've reached the carrying capacity.
Yeah.
So
the excess tigers. Yeah, yeah.
From there,
correct.
What would be the issues at hand? Would you come to it?
Yeah, no, so basically the thing is that genetics is only one part of the solution. So a lot of individuals which are spilling out of protected areas tend to be dependent more on cattle. Now, for example, a population like Simlipal, there is no tourism, very well protected. There are no cattle inside. So those tigers are very wild, if you want to say that way. Their diet is very wild. And now if you're going to take a behaviorally very different individual and put it into there, that might have other problems. But those things we don't know the answer to. What I think might be best is to create temporary populations where individuals mate and then allow them to naturally disperse so they integrate into those populations. But yeah, that is
So
Odisha is empty. Yeah.
they're kind of connected to the forest in central.
They are connected, very much connected, but I'm going to talk about that now. So basically, that is the first question, right? So this is Simlipal, and these are all those other populations we talked about, Kanha and Pench and all that stuff. So we just basically use those resistance values and threw them on this landscape and said, what is the probability given some kind of a dispersal threshold that animals will actually reach Simlipal? So if the dispersal threshold is 200 kilometers, that's the average dispersal, they'll never reach Simlipal. If it is 500 kilometers, they will with a low probability. And only if they are dispersing 1,000 kilometers on average, they'll get there. So it's basically very far, right? It's isolated, not necessarily because there's no forest cover, but it's really far. And basically, there's a lot of hunting in Chhattisgarh, Jharkhand, and Odisha. And so basically, there's very low prey base. So there's no place for them to stop and then maybe move on or whatever. Okay, so basically, to summarize, actually, I should correct myself here, because just recently a tiger has shown up here. It's disappeared again, but it's like these events are going to be really rare, but maybe they will happen. Okay, so we also just did some very simple simulations to ask how could the frequency have been so high in Simlipal and so low outside? And this is basically as a function of the time, since there was this bottleneck. We know that Simlipal went through a bottleneck. There was a lot of insurgency and a lot of poisoning of salt licks because a lot of animals were killed. Or was it that the bottleneck size was low? So basically the answer is you have had to have a very strong bottleneck with very few individuals. Only then, if you had 2 or 3 individuals left, that you could actually get this big difference in mutation frequency. So basically what we're doing now is, again, we're trying to make this a long-term study. This was from 2017-18, and we just went back last year, 2023, and we collected samples again. And so we expect just by population genetic theory that the frequency of this mutation will keep increasing. The probability of fixation is the probability of its frequency. So it should increase and get fixed. So the question is, has it increased? We don't have the answer yet, but from camera trap studies, where they're camera trapping black tigers here, the number of black tigers has gone up to half the population, which indicates a frequency of 0.75. Okay, 0.75 squared is half. But we'll see. We're kind of working on this. And we're hoping to do more work in Simlipal if we are able. So very quickly, I don't know how much time I have. Okay, I have some time. So about Ranthambore, where again, we've been working for a very long time. Isolated population. I love this picture. There's a famous tigress called Arrowhead. She probably has an Instagram account. I don't know. But this is a hunting lodge. So you may be surprised to know that most of our protected areas in India are either around temples or hunting areas. So this is where people used to hunt. And this lodge is where the hunters used to stay. But of course, today, the tigers rest there in the heat of summer. So of course, this is like, it's amazing to have worked with these people. And Anubhav actually just recently got a job, faculty position at IISc. So really excited to, hopefully we can continue to work with him. So basically what he did was, he said, okay, small isolated populations should be inbred. So individuals in these small isolated populations should show a higher inbreeding coefficient or more homozygosity in their genome. And that's what we found when we compared tigers from Rajasthan to central India, which is larger and connected, and southern India. No. Oh, no. Inbreeding depression has nothing to do with depression. Inbreeding depression is the loss of fitness, reduced reproduction, reduced survival because of inbreeding. And that is because we all share, like related individuals share bad mutations. And when we mate with relatives, they come together, and that causes inbreeding depression. This is a theoretical and an empirically shown paradigm in mice, in humans, and so on. But we don't know what inbreeding depression looks like for tigers. We haven't seen it yet. We know there's inbreeding. Individuals are related. There's high shared homozygous stretches, but we don't know what it's going to look like. So just very quickly go through this stuff. Individuals with higher inbreeding also have more damaging mutations. So, basically you have less homozygosity, or you have less variation. You also have more bad variation. This is also true for loss of function mutations, basically kind of badder bad. But basically, the other thing which we found is that there's also signatures of what's called purging. That is, Ranthambore actually has fewer very bad mutations compared to central India. But all the mutations that it has, which are very bad, are homozygous. That means every individual has 2 copies of them. So that means it will experience it. But some of them seem to have been, the number seems to be less. So this is a process, this is thought to be statistically what's called purging. That is, immediately when you have a bottleneck, all the very, very bad mutations, the animals which have them, they just die. So their frequency goes to 0, right? So how can we find out what was purged? The only way to do that is to go back in time and to look at variation in the past and see what has actually gone from low to 0. Very ambitious, may not work out. Anyway, so this is another crazy wild goose chase. This is me climbing a ladder in the Maharaja of Sarguja's palace. And you can see he has this trophy, a tiger shot in Chhattisgarh in the early 1900s by his grandfather. And Chhattisgarh tigers are extinct today. So hopefully, if we can get genetic material out of this, he was very particular that it should not be shared. So we can try and understand more about what did we just lose habitat and numbers, or did we actually lose genetic variants and so on. So that was like still statistical stretches of homozygosity in the genome and all. It's nice, but when you think of inbreeding, you mean mating between relatives, right? So what is that? Do we see that? So to explore that, and this is Aditi, she just did her pre-synopsis seminar yesterday. This is all her work. We are trying to reconstruct the wild pedigree in Ranthambore. Why Ranthambore? Isolated population, no one coming in or going out. So it's a good control site. And how do we do this? Now here, we can't just walk through and collect any scat. We want to know who is the mother of who is the father of who. So we need to collect mothers, their offspring, and possible fathers. So we need to follow individuals, right? So this is basically what we do is individuals are identified by their stripes. We have all numbers for them, not we, the forest department. So we track individuals. There's a huge guide network in Ranthambore, and they're all our friends, and they tell us, T16 wahan dikhi, etc., etc. So we zoom in there in our Jeep and basically watch. And we never knew this before, but when tigers get up, they're sitting over here, there's tons of hair on the ground. So basically, Anubhav actually showed that with this hair, you can actually sequence genomes. And that's what we've been doing. We've been collecting these shed hair samples. And this is basically, since we're thinking about it in terms of mothers and their children, they are matrilines, right? Mothers and their children and grandchildren and so on. So Ranthambore is thought to have 1, 2, 3, 4, 5, 6, 7, 9 matrilines. And the first question we wanted to ask is, are all these matrilines different from each other? And can we sample all of them? So basically, this is just a representation of, you can see that the females are here, the cubs are here, the mates are in white, because we need to assign them. We don't know who the fathers are. Mother and offspring, we see them together. So we can assume and then test with genetic data, right? So basically, it's just doing paternity analysis for wild tigers. So what did we find? Yes. So this family is very successful. This family, sorry. I should not use this at all. This family, right? This one. Yeah, this one and this one to some extent, right? So this female is T16. That is Machli. She is a very famous tigress again. She was extremely aggressive. She had a super territory and she had tons of offspring. So she is very successful. She was very successful. Yes.
Also super
no, super. Sorry, I don't mean it in a bad way. She was very good at defending her territory. That's what I mean. Like she was, and she would always attack tourists. So I mean, not attack, but like tourists love to see her. People would come to see Machli to Ranthambore. She has earned more than $1 million in tourism revenue. So she's really famous. She has a Wikipedia page. So anyway, so yeah, some of the lineages have very few individuals, right? So first of all, the first question was, maybe these are all very different and, these are the founders of Ranthambore. The last kind of decline was in the early 2000s, and all the individuals that are there today are just descended from these matrilines. So, yeah.
These four.
Yeah.
They're very small.
Very small.
They have very big territory.
Yeah, so a lot of these are in the tourism zone, and there's a big lake there, and there's a lot of prey there. Some parts of Ranthambore are very dry, and there's more of a desert, like chinkara and all these animals, low prey density. So these are not ideal, these are often in non-ideal territories. Okay, so this is not a nice figure, it's just raw, so please excuse the graphics. One family, like matrix, we could not sample at all. We could never see them, and they're all dead, they're out. But all the others that we sampled basically fell into two, this is the whole mitochondrial genome, because matrilines are, you get mitochondrial DNA from your mother only. So basically there are two matrilines. These are all very similar, like one or two differences, and these. And basically these big families you talked about are very close to each other, like probably sisters or something like that, very close to each other. And all the chotu families are basically this other matriline. But even these are not very different. There's just some 6 or 7 mutations different between them. So they're probably not, they're all very similar to each other. So that again suggests that the founding population itself was all related to each other. And this is the great, like, this is the new results, which I'm really excited about. So this is crazy. This is 23 mate pairs finally assigned, and 65 individuals' genomes, sampling since the last 8 years. And what you can see are, there's all kinds of matings. These are all matings. Basically, there are a lot of uncle, niece, aunt, nephew matings. There are 2 half-sib matings, but there are no sib matings or mother-offspring matings. So there is inbreeding, but it's not first-degree relative inbreeding. So that's interesting. Now, whether that is by chance or something, yeah, it could be by social structure. Males usually disperse away from where their females, daughters like push out their mother or get a similar territory close by. We don't know, but this is the first verified like paternity or family tree for tigers, wild tigers. So we'll see.
And this is what my sampling is here.
Yes, not only hair, we also have some blood samples here. So every time the forest department catches animals, we have a permit, they give us blood with it. Yeah.
MHC complex
Yes,
push to get heterozygosity.
yes.
I mean, this is a population in which then that sounds like there might be something similar going on to drive heterozygosity up as best as
Yes.
it can under
So those
constrained
things,
circumstances.
yeah, we have to test. So first we have to have the pedigree, and then we have to test those things. There are many other things I would like to test. For example, segregation distortion. So we said there are lots of bad mutations, but we don't really know whether they are bad. So we can actually look at whether they're being inherited in a Mendelian fashion as expected by the pedigree. So we can try and identify mutations which are potentially not being inherited and then look for their functional effects. Something we're hoping to do in the future, collaborating with making cell lines and actually making mutations and testing it. Again, very ambitious, may not work out, but this is the base on which we can now do many studies, and hopefully we can continue to build this pedigree. What we do now, it's easier. Every summer we go, there are new offspring, so we sample them and then we keep building like that.
Could the absence of these extremely close inbred, like full sibling that you mentioned, be just that such offspring are not viable or something? Maybe.
Exactly, yeah, they could be, right? So for example, now the other thing to look at is inbreeding depression is supposed to cause decreased fitness, right? Or basically reduced reproduction or something like that. So we do find, for example, that interbirth interval in this population is increasing. That is, females are having offspring. They should, normal tiger physiology says once in 2 years or 3 years. It seems to be up to 4 years. And animals, the cubs are not even moving out from their mother until they're 2.5, though they usually leave at 1.5. But I don't know whether that's because of density dependence. That could also be, density dependence could also be driving that, or is it genetics? Like who's related to who and so on. But I think what you're asking is, there are also individuals who are not reproducing. Now, we don't see the cubs, but they could mate and the cubs could have died. They could have had stillbirths. We don't know that in the wild. It's very difficult to assess that. So that's something we're taking up in zoos, where actually in Indian zoos, they do a lot of inbreeding. I don't know why. So,
Like, is that intentional or?
I mean, it's just not scientific. They're just not thinking about it. So basically, we can actually look for these stillbirths or abnormalities, and I'll come to that. But yeah. Okay.
The audience to just stop right now
I
with
just quickly
questions and
finish.
let her
Yeah.
finish and then we'll take.
Yeah. So basically, also, if indeed there's an isolated population, there's mating between sort of relatives, we should expect inbreeding to be increasing over time, right? So we don't have too much data yet. Basically, we have individuals, this birth year. So individuals born more recently should be more inbred, right? Because there's nowhere else, there's no inflow. So basically, we do find a very weak correlation, but there is an increase. Like the individuals today have almost 55% of their genome is homozygous. This is crazy high. Like even in Ashkenazi Jews or known inbred populations of humans, that sounds bad. Consanguineous populations of humans, it's like 0.3, right? The proportion of the genome which is inbred. So this is really high. Okay. So, of course, the big question is, so what? What is this inbreeding going to look like? We have the genome, don't you know? Can't you figure it out? Like you have all these mutations, you have an annotation, like which part of the genome is which gene and so on. Can't you say what inbreeding is going to look like? You may want to ask me. I ask myself all the time, and I don't think we can yet. I have a hunch, or I don't know. I think that there may be syndromes of inbreeding. So if you look at a lot of data from cats, a lot of inbred cats have eye conditions. They have hip conditions. They have heart conditions. So why only those? Why not have any other kinds of things? So I don't know the answer, but for example, this snow leopard, we're just trying to get these data. Snow leopards in US zoos, there's a very strong this phenotype called ocular coloboma, where the eyelids don't close. So it's like a half eyelid, like this. The developmental thing. So associated with inbred individuals. So if we can actually get those data and analyze it, find, like with the black tiger, find potential mutations, we can try to understand, maybe are those there in the Ranthambore tigers? And also surprisingly, and again, completely anecdotal, one of the females, which is the litter of the half sib mating, has an eye deformity and she has not mated. So again, circumstantial, who knows? But this is something which we're hoping to follow up in a combination with zoo data, wild data, and maybe some bioinformatics kind of approaches. Okay, we've also done some work on other species, not obsessed only with tigers, generally big cuddly things, but not only tigers. So we've just started working on lions. Asian lions are a quintessential example of bottlenecks and inbreeding, but nobody has studied them. For the longest time, I was even scared to think about it. I always wanted to do it. But finally, after 25 years, I've been wanting to do this for that long. Applied for permits to work on, because getting permits in Gujarat is close to impossible. And we've actually managed to sample, again, shed hair. That's the key. If you ask for blood, they immediately say no. But lions from Gir and surrounding areas have something like 70% of their genome in homozygosity. We also saw in these animals, which are actually wild-caught animals in the zoo, neurological disorders, eye conditions, shaking disorders, hip dysplasia. So maybe, there are potentially inbreeding effects. So let's see, maybe we can find out more about this. And hopefully, as Sandhya was suggesting, think about models for genetic rescue. Now, in the case of the lions, the only possible source is Africa. So we know how all that went down. So yeah, but still, that's something which we need to think about. We have some really a paper which is almost accepted on elephants, which is really cool, showing that elephants colonize India from the north. And as they go from north to south, there's a serial dilution of diversity. And so they have less and less variation as they go from north to south. So there's a lot of geographic patterns as well. So that's kind of more exploratory. But anyway, that's about it, I think. Yeah, so this is just to show you that with the lions, they have long black hair, the mane and all that. So anyway, again, we're sampling shed hair and we are sequencing genomes from it. It would be nice if we could get blood, but very difficult in India. So I'm really lucky to work with lots of amazing people. There are many more people here than in my lab. Yeah, and thank you so much. I don't think I could have done any of this work in any place other than TIFR. So thanks.
Wonderful colloquium. Absolutely. Everybody is spellbound with the data, the results. So yeah, questions? Yeah, Mitesh.
So normally in biology, we do other way around, like we pick some animal model, we study that, and then we try implementing that on human. Can something be done on a opposite direction, that we have information from human developmental diseases and all,
Yeah.
and we apply that instead
Yeah.
of?
Yeah, we're doing exactly that. So basically, there's this thing called a PLE score, which is like a mutation if it is in very low frequency, because we know so much about human populations and how the frequencies of variation should be. If it's substantially low, it's probably under negative selection. That is, it's kind of been removed as much as possible. So we know potentially that is detrimental. Then there's this ClinVar database for mice, where there's so many inbred mice lines. So we know phenotypic effects of specific mutations. And then we also have bioinformatic approaches and Go analyses. For example, this, whatever, pathway analysis to say, okay, this potential gene might have mutations which are bad, right? This is a gene which could have things which are bad. So we're trying to look at that. So I didn't talk about that. We're looking at how our mutations overlap with those. And we've just actually found some very interesting specific mutations which we find in homozygous state in stillbirths. But we also find what in humans, knockouts they're called, right, would cause mortality. We find tigers happily walking around with those genotypes. So
that will be another interesting thing to look at why it is not lethal there.
Correct. So yeah, so we just kind of crazy to do genetics with a non-model system,
understand,
but we're trying.
Amazing work.
There's a question here.
Yes,
Yeah, so I was just wondering, Uma, I don't know if it's possible to overlay on this an epigenome map. For the reason I was asking is, as you increase inbreeding numbers, are you going to associate this with epigenetic changes that are reflective of hastened aging, accelerated damage / is there a way to overlay it just to use that as, because I mean, obviously it's going to be difficult to figure out whether you have a fitness cost necessarily, but is there a way to do that? I don't know if that's possible.
We can try, but for that we can use probably blood samples because we need good quality DNA for that.
I guess you can do that from circus or / the zoo populations at least.
Yeah. And we do have many Ranthambore tigers blood also. So we can try, but right now, it's a lot of this work is expensive. So I need to probably raise some more money for that. But yeah, that's a very good idea. You're talking about cats. So yeah, so basically, that's something again I'm trying now to get in touch with. There's a lot of people who, like with dogs, now there are services. You can have your dog genotyped for specific like dogs have, like Cocker Spaniels have ear issues, things like that. So there are known mutations which cause disease phenotypes. The work is much more in dogs than in cats, but that's something which we can try and look at.
Okay.
Yes. Okay, I have two. One is that, maybe the reason for extinction could be inbreeding, maybe at the later stage.
Yes, definitely.
But when the population starts declining, that may be for a different reason, isn't it? Because you have varieties.
Definitely.
So you have seen that
so I totally agree with you that I think habitat, fragmentation, protection, prey base, so ecological things, nitty-gritty of survival of animals is way more important than genetics. I'm probably looking at it from a fundamental research perspective. So I'm not going to oversell that this work is going to save their survival kind of thing, but it is one of the components. Definitely, that's more
the
important.
question. I'm just curious to know that, when you were looking at samples and so on, did you find any sample where, some species came out of tiger mixed with some other animal or something like that?
No. Not yet. Luckily, we can very easily distinguish these species from each other. And the thing is, we have a lot of statistical power because we're using millions of markers. But there are definitely hybrids. So for example, in Indian zoos, there are hybrids between Asian and African lions.
Mhm.
Now, we have not looked at it, but
I see.
yes, of course. And in fact,
Naturally also doing that.
no, we haven't found anything like that naturally. We have actually, we just reported a hybrid between, for the first time, between a wolf and a dog close to Pune. So this was like a wolf which looked odd with the wolf pack, and some collaborators who have an NGO sampled it, and when we looked at it, it was actually a hybrid. But we have not found hybrids between lions and tigers. Ligers are interesting because they actually are size indeterminate. They keep growing. Don't know why that is, but they're interesting for that reason.
And polar bears are in the wild.
Yeah, because of habitat.
Okay. Thank you, Ramesh. And we can continue the conversation while having snacks. But thank you, Uma. This was fantastic.
Yeah.
And thank you, Sandhya, for bringing her here and recommending her. Thank you very much. We thoroughly enjoyed. So all of you are welcome to join for some snacks, and we can have some more time with Uma.